Hello everyone. So today in this video we are going to solve the January 2020 biology paper 2. Let's start. Now I am not going to read the entire passage. You can read it in your home. I will just solve the questions related to the passage. Question number 1. Explain why cattle with damaged lungs have gas exchange problems. It is extracted from lines 2 to 3. If you go back to the lines 2 and 3, this is basically the first 3 lines of the passage. The bacterium Mycobacterium bovis causes the lung disease bovine tuberculosis in cattle. Infected cattle have damaged lungs, which causes gas exchange problems and makes them cough into the air. So in question number A, it wants you to explain why damaged lungs causes gas exchange problems. We know that damaged lungs means the alveoli will also be damaged, and damaged alveoli causes or damaged alveoli have lower surface area, and lower surface area means that the rate of diffusion decreases, which eventually leads to the gas exchange problems. So let's write it down. Because they have damaged alveoli, which have lower surface area, so the rate of diffusion decreases. Leading to gas exchange problems. Moving to question number B. Explain the advantages of drinking pasteurized milk rather than unpasteurized milk. Lines 4 to 5. The pathogen spreads to humans if they inhale air containing the bacterium. Humans also become infected if they drink unpasteurized milk from the infected cattle. Milk is usually pasteurized by heating it for a period of time at a high temperature. So it has already mentioned that during pasteurization, the milk is heated in a high temperature. We all know that high temperature obviously would destroy the bacteria, and as the bacteria inside the milk are already destroyed, so there is no chance of spreading the infection or spreading the disease. So let's write it down. The high temperature used for pasteurization of milk, the high temperature used for pasteurization of milk destroys the bacteria so there is less chance of spreading the infection. Infection. Part C suggests 2 reasons why there were fewer cases of bovine TB in Europe compared with outside Europe. Lines 7 to 9. Bovine TB can kill humans. In 1 year. 13,400 people died from a total of 149,000 reported cases of TB. Some of these cases were from Europe, where 103 people died from 1,290 reported cases. The rest of the cases were from outside Europe. So we can say that in Europe there were only 103 people died, and the total number of cases for TB was 1290, which is lot less compared to the entire world. This is because a fewer number of people stay in Europe compared to the entire world. And maybe the cattle were vaccinated in Europe, or the cattle were screened before, or they were killed whenever they were detected with the bacteria. So we have to only write 2 points for this. So let's write the 2 points. Cattle are screened in Europe, and the other reason would be fewer people in Europe than rest of the world. Fewer people in Europe than rest of the world. D. The case to death ratio in Europe is 12.5:1. Calculate the case to death ratio from outside Europe. So here the total number of cases in the entire world, including Europe, in the entire world, and this also includes Europe, including Europe, was 149,000, and the total number of cases inside Europe, total number of cases inside Europe was 1290. So we can now know the number of cases outside Europe was, or would be, 14900 minus 1290. Let me draw this or write this on the blank pages so it would be easier for me to explain. So I'm moving to page number 23. I'm doing the part D of 1 here, the working, I'm showing it in here, and the explanations for this, for the working that I chose. The total number of cases in entire world, and this includes Europe, that was 149,000. Total number of cases just inside Europe was 1290. So we can now know the number of cases outside Europe would be 149,000 minus 1290, and the answer is 147710. These are the number of total cases outside Europe. And now we need to find state the total number of death cases due to TB across the world. Total number of dead cases, that was 13400, and total number of dead cases inside Europe was 103. So the number of dead cases outside Europe would be the number of dead cases outside Europe would be 13400 minus 103, and you get a value of 13297. So the total number of cases outside Europe was 147710, and the total number of dead cases outside Europe was 13297. Now let's move back to the page 4. I'm gonna rub all these, and now we need to find the ratio of death case to death ratio. So case is to death. The total number of cases outside Europe was 147,710 divided by total number of death outside Europe was 13297, and if you calculate it, you will get a value of 11.1:1. So this is the ratio for total number of cases is to death. So this is 11.1:1. And if you want to see the working, let me go back to that page again and zoom out. So this is the working for the part D. I hope part D was clear. Moving to part E. Territorial behavior reduces the spread of disease between badger groups. Lines 19 to 20 suggest two other ways that badgers benefit from territorial behaviors. We all know that because of having territories, there will be less competition for food and for mates. So these are the two reasons, and they also asked for two reasons. So let's write it down. Less competition for food, competition for mates. Explain how vaccination reduces the risk of infection in badgers. Line 24. At first, we need to define that how vaccines actually work. We all know that vaccines are actually made up of attenuated pathogens that contain the specific antigen that would trigger the immune system to produce antibodies and memory cells. The antibodies would destroy the antigens of the attenuated pathogens inside the vaccine, and the memory cells will go and store or get stored in the lymph nodes, and they provide immunity. How they provide immunity is that whenever that same antigen reinfect the body, the memory cells came out of the lymph nodes and come into the site and destroy the pathogen before it can even cause minor symptoms of the disease. So let's write: vaccines cause attenuated pathogens that contain antigens. Vaccines use attenuated virus pathogens that contain antigens which trigger the immune system to produce memory cells to provide immunity. They act as a secondary immune response which produce antibodies faster when the pathogen re-enters the body. So this is the answer for part F. Moving to part G. Describe how scientists could find out if badger vaccination reduces TB in cattle. So whenever we need to find out if one treatment or one solution is effective or not, we need to always take two different observations. One observation containing the treatment and another observation not containing the treatment. So here, as we need to justify that the vaccination of badgers is useful or not, so we need to take two different fields of the same size and area, and we need to use the same number of cattle in both fields, and then we need to place same number of badgers in the both fields, but we need to use the vaccinated badgers for one field and unvaccinated badgers for the another field, and then count the number of cattle with TB in both fields. And by counting the number of TBs, or sorry, but not by counting the number of TBs, by counting the number of cattle with TB, we can find out how effective the vaccination for badgers is actually. So let's write. We need to use two different fields of same size and area. Not we, that scientist actually, because the question asked how scientists would do that. So it's better to use scientists. Scientists need to use two different fields of same size and area and put same number of cattle in them. They need to use vaccinated badgers for one field and unvaccinated for another field. And then count the number of cattle with TB. Now always make sure that you never mention that what the results are, because we don't know that if the results would be positive or negative. So never ever try to write the results of any investigation. Just write the procedures and never the results. And with this, the question number 1 ends. Moving to question number 2. Transpiration is the evaporation of water from the surface of a plant. Explain why transpiration is useful for plants. We know that transpiration is actually a pool which takes the water, or which actually pulls the water from the roots up to the leaves, and without these pools the water would not travel or against gravity, because gravity acts downward. And for this, as water moves up to the leaves, water also carries specific minerals required for plant growth like magnesium, potassium and nitrate. They're all mixed in the water and they come along with the water and reach the leaves, and then they reach their desired destinations where they are required. And also water is required for photosynthesis inside the leaves and obviously they get the water inside the leaves by transpiration pool or due to transpiration pool from the roots. So now let's write it down. It pulls water up to the leaves so they can be used for photosynthesis. They prevent wilting and help in transporting materials. And help in transporting minerals. Moving to part B, a student uses the apparatus to measure the rate of transpiration in still air and the rate of transpiration in windy conditions. The student calculates the water loss of loss of water after 1 hour in mg per centimeter cube of leaf area into 10 to the power -4 for the plant in still air. He then repeats this calculation for the plant in windy conditions. The graph shows the student's result. Water loss after 1 hour in mg per centimeter cube of leaf area in 10 to the power -4. So let me first write a unit for this y-axis quantity. It says water loss after 1 hour in mg per centimeter cube. So the in unit would be mg divided by centimeter cube and h, because it is the water loss after 1 hour. So it is basically water loss per hour. So that's why we can write mg divided by cm square h. The total leaf surface area for which transpiration occurs is 250 centimeter square. Calculate the water loss from this plant in still air in mg per hour. So we need to just find the water loss mg per hour without the area. And now from this graph we need to find the value for mg per cm square h for still air, and that is, we can see that it comes between 21 and 22. So we can obviously conclude this as 21.5. So as 21.5 has a unique unit, that is 21.5 into 10 to the power -4, because it was already on the y-axis, and the unit for this would be mg/cm square h. And as we need to find the water loss in mg per h without cm square, so we need to just multiply the 250 here, 250 cm square. Now why I am multiplying the 250 cm square, I'm going to show a rough work for this. Let me move to page number 22 for this part. Now the water loss per hour per centimeter square was given by the equation mg, or given by the unit of mg cm square h. We can obviously write it as mg cm square h. And if I multiply the total area of the leaf, that was in again cm square, we can see that this cm square and this cm square cancel each other out, and the leftover answer would be, or would have the unit for mg divided by h, which eventually stands for mg per hour. And that's why I am multiplying the total area of the leaf with the 21.5 into 10 to the power -4 value. So let's move to the page where we were. So now if you calculate this, you will get a value of 0.5375, and this will eventually have the area of mg /hour. So it will be 0.5375. Moving to part B, explain the results shown by the graph. So let's look at the graph for once. We can see that in still air, the water loss was 21.5, and in windy conditions, the water loss was 25. So we need to explain these results that is shown in the graph. We all know that in windy conditions, or whenever there is wind present, the wind will always carry out the water from the plant surface, and as it will carry out the water from the plant surface, it will cause or create a concentration gradient which causes more water out of the plant by evaporation. So let's write it down. Water loss from plants, in windy condition is more than still air, because wind moves water away from the plant source so it maintains a concentration gradient which causes more water move out of the plant by evaporation. So with this, part B2 ends. I hope part B2 was clear. Moving to part C. Justify two factors that the student needs to control to make a valid comparison of transpiration in still air and in windy conditions. So in order to make it a valid comparison, we need to control few things like the light intensity and the temperature and humidity. So why light intensity is a factor that should be controlled in this comparison? Because light intensity affects the stomata opening, and from this stomata opening, water actually comes out of the leaves. So light intensity affects the transpiration. And temperature always affects the rate of evaporation, so we have to control the temperature for the both plants to make it a valid comparison. So let's write it down. Same light intensity as it affects stomata opening and same temperature as it affects the rate of evaporation. Evaporation. And with this, question number 2 ends. Moving to question number 3. The human heart pumps blood when its muscle tissue contracts. Diagram 1 shows a human heart and a cross-section through its ventricles. Which of these statements about the ventricles is correct? The left ventricle wall is thick because it has to pump blood to the body. So we need to justify that which out of these four statements, which one is correct. So the first one says that the left one ventricle has a much thicker wall compared to the right ventricle, as the left ventricle has to pump blood to the entire body, which is actually correct. So let me say it's correct. The left ventricle wall is thin because it has to pump blood to the lungs. We all know that left ventricle, whenever it contracts, the blood moves to the aorta, and aorta does not go to the lungs. So this one is wrong. The right ventricle's wall is thin because it has to pump blood to the body. The half of the statement is correct, that the wall of the right ventricle is thin. This is correct, but the reason for this half statement is wrong. That is, they give that it has thin wall because it has to pump blood to the body, but we know that whenever right ventricle contracts, it pumps blood to the pulmonary artery, and pulmonary artery leads to the lungs. So it is wrong. Right ventricle wall is thick because it has to pump blood to the lungs. Again, half of this statement is correct, but the other half is wrong. And here it says that the right ventricle wall is thick, which is not thick, it is thin. So that's why this is also wrong. So out of the four statements, only option A is correct. So we need to just select option A. Give it a cross mark in the box and rub it out. Part A is done. Moving to part B. Ventricle muscle tissue is damaged if ventricle muscle tissue is damaged if the blood supply is reduced. Diagram 2 shows the damaged part of the human heart with a cross section through its ventricles. Explain the cause of the change in the appearance of the muscle tissue in diagram 2. We can see that the left ventricle has, or the lumen for the left ventricle has become a lot bigger, which eventually indicates that there has been a blockage in the coronary artery, and due to blockage in the coronary artery, the muscles or the heart muscles cannot get enough oxygen in these regions, and due to lack of oxygen, they cannot respire aerobically enough to produce enough energy, and therefore the cells in these regions die out, increasing the lumen for the left ventricle bigger, or increasing the lumen for the left ventricle. So let's write it down. Ventricle wall is thinner due to blockage in coronary artery. Ventricle wall is thinner due to blockage in coronary artery. So less oxygen is transported to the heart muscles. The heart muscles die due to less aerobic respiration. So with this, part B ends. Moving to part C1. Scientists are investigating the use of stem cells to repair damaged hearts. The stem cells are injected into the ventricle muscle tissue. Explain why stem cells could repair a damaged heart. We all know that stem cells are unspecialized cells. They can differentiate by mitosis to form any type of cells. So they can also form the muscle tissue for the ventricles. Let's write it down. Stem cells are undifferentiated or unspecialized cells. They can differentiate by mitosis to form ventricle muscle tissues. Muscle tissues. So moving to part 2, the stem cells are obtained from the person who has the damaged heart. Explain why using cells from the same person is better than using stem cells from a different person. So we all know all cells have the specific antigens, and cells, all the cells from the same person will have the same antigens, so there is less risk of rejection. So, and there will also be no immune response, and there will be less risk of infection too, because the stem cells from another person will obviously contain the diseases of another person. So if a person is using another or someone else's stem cells, they will also get the diseases of another person. So that's why using stem cells from different person can cause or increase the risk of infections. Let's write it down. All the cells from the same person have antigens so less risk of rejection, there will be no immune response, and no risk of infection. And with this, our question number 3 ends. Moving to question number 4. DNA and RNA are involved in the process of transcription and translation. State 3 ways in which the structure of a DNA molecule differs from the structure of an RNA molecule. So we all know that DNA is a double-stranded helix, or it has a helix structure. DNA has thymine, whereas RNA has uracil. DNA has deoxyriboribose. So these are the 3 differences between DNA and RNA. So let's write it down. DNA has double strand, DNA has thymine, DNA has deoxyriboribose. Deoxyribose. So these are the 3 points, and with this part A ends. Moving to part B, describe the differences between the process of transcription and translation. So this is actually a more memorized answer. You should actually memorize the answer for this specific questions, because these are really common. So let me first explain it to you. The transcription is a process by which mRNA is made by copying the, or not copying, by copying the nucleotides on DNA, and it happens inside the nucleus. And translation is a process which happens on the ribosomes in cytoplasm, and they're involved with mRNA and tRNA, and in this the anticodons binds with the codons and it forms polypeptides, the primary structure for proteins. And these are the these are the differences for these 2 processes. Let's write it down. Transcription occurs in the nucleus. And translation takes place at the ribosome. Transcription uses DNA, whereas translation uses mRNA and tRNA. In transcription, DNA unzips and in translation, anticodons binds to codons. Bind to codons. Transcription produces mRNA and translation produces polypeptides. Produces mRNA and translation produces polypeptides. And with this 4B ends. I hope it was clear. Moving to part C. The DNA molecule codes for amino acids used to make proteins. There are 4 different bases in DNA and 20 amino acids used to make proteins. Use this information to show that a minimum of 3 bases in the DNA molecule is required to code for 3 amino acids. So I need to explain it to you. So I'm gonna show this or explain this in a blank page. So as it was already stated in the question that we need to show each amino acid need to have 3 bases. So and there are 20 different types of amino acids. So as one amino acid, let me zoom it out. One amino acid consists 3 bases, 20 amino acids should consist 20 into 3, that is 60 bases. Now as there are 4 different type of bases in DNA, now the 4 different type of bases will arrange itself in different formats, so they will create at least 60 bases. So now number of bases formed, if we consider it forms like in 3 bases per amino acid, so it will be if red as 3, then it will be 4 into 4 into 4. Each base comes in 3 times but in different orientation forms the amino acids. So it will be 64 bases. which is obviously greater than 20 total number of bases in 20 amino acids, which indicates that the 64 bases, we can arrange the 64 bases in different orientation to make 20 combinations for the 20 amino acids, for the 20 different amino acids. Now if read as 2, like if they were like 2, like 4 and 4 bases only, that is if read as 2, it would be 4 and 4, and this will be eventually 16 bases, which is a lot less than the total number of bases required for the 20 different amino acids. So the amino acids cannot have 2 bases, they have to have 3 bases. So this is the entire explanation for 4C, and that's how you actually have to arrange or write the answer to. I wrote it in here because it took a lot bigger space, and it was easier for me to write in a blank page than on the lined margined pages. So I hope 4C was clear. Moving to question number 5, because with this question number 4 ends. Moving to question number 5. The diagram shows part of a human nephron with parts labeled P, Q, R, S and T. What is the name of the part P? So this is part P, and we know it is collecting duct. So this is option A. Where does ultrafiltration occur? We know ultrafiltration always occur in the glomerulus. That is option T. So the answer should be option D. Where is permeability affected by ADH? So we know that ADH actually makes the collecting duct more permeable, and we knew that collecting duct was P, so the answer again would be A. Where is glucose selectively reabsorbed into the blood? That is the first convoluted tuboil, and that is R. So R is in option B, so answer is option B. Moving to part B1. Scientists investigate the relationship between birth mass and the number of nephrons in the kidney. The scatter diagram shows these results. Describe the relationship shown by the scatter diagram. So I'm not going to say that it is a linear graph, because as it is a scatter diagram, the points are not approximately or not even exactly one after another, and they are like scattered in an uphill pattern, like they were going upward. So I'm gonna say they have a positive correlation, because this entire scatter diagram indicates or has an uphill pattern, and therefore as it is a positive correlation, indicates that the birth mass has an effect on the number of nephrons. So let's write the answer. It shows a positive correlation. Correlation. Increasing the birth mass is associated with more nephrons. And with this, part B1 ends. Moving to part B2. Babies born with low birth mass are more likely to develop high blood pressure when they become adults. Explain why protein in the urine of these adults may be an indication of kidney damage caused by high blood pressure. So we all know that proteins are a big molecule, and during the ultrafiltration that happens in this glomerulus part, during the ultrafiltration, only the blood plasma passes out into the Bowman's capsule, and the glomerulus holds on to the bigger molecules such as blood proteins, proteins, and most of the glucose and other bigger molecules. Now, in normal urine sample from normal patients, they will not have protein because their Bowman's capsule is perfectly fine. So they won't obviously allow proteins to pass through them because they don't have enough pores that are enough big for proteins to pass through. But due to high pressure, for the people with for the people with the disease, due to high blood pressure, the protein molecules will be forced to pass out of the glomerulus and thus damaging the glomerulus, because they will make large holes in the glomerulus, or the openings or the holes of the glomerulus would become more big, and this will obviously causes other problems in the human body also. So let's write the answer. The answer would be proteins are large molecules. They normally will not pass out of the glomerulus. They normally will not pass glomerulus, but due to high blood pressure, I'm going to write it as BP, protein molecules are forced out of the glomerulus, therefore damaging the glomerulus. But high BP forces proteins molecules out, damaging the glomerulus. And with this, part B2 ends. I hope it was clear. Moving to part B3. Describe a simple test to show that a urine sample contains protein. We can just write the test for protein, that we will add burette to the urine sample, and if it turns purple, then it indicates that that protein is present. So add burette into the urine sample. And if it turns purple, then protein is present. And with this, our question number 5 ends. I hope it was clear. Moving to question number 6. The diagram shows a fermenter used to grow microorganisms. Describe the function of all the pedals in the fermenter. So this is the pedal of the fermenter, and we know the pedal is here. It acts as a stirrer, like it stirs the mixture so that it has a constant temperature throughout the entire mixture, and oxygen is also distributed equally throughout the entire mixture. So the answer is here to distribute heat and oxygen constantly or thoroughly throughout the entire mixture. So answer is to distribute heat and oxygen thoroughly entire mixture. Part B, explain why the fermenter has a filtered air supply, because we all know that the air supply from the outside sources will always contain microorganisms, and if we are going to use as we are going to the main idea for using the fermenter is to see the growth of microorganisms of a specific type. So now if you are gonna use air from a different source, so we always have to use a filter so that no other types of microorganisms from the air source can enter inside the fermenter. And because if microorganisms from other source enter the fermenter, it will cause competition for oxygen and therefore competition for aerobic respiration, and the microorganisms that we are trying to produce will obviously face competition and they might die. So we do not want that, and that's why we filter out the air. So let's write the answer. Filter removes bacteria to prevent competition for oxygen for aerobic respiration. Filter removes bacteria to prevent competition for oxygen for aerobic respiration. And with this, part B ends. I hope it was clear. Moving to part C. Explain why the temperature recorder is connected to the liquid in the fermenter and the water input in the cooling jacket. So it wants explanation that why this temperature recorder is connected to this water cooling jacket. So it is connected here because we can monitor the temperature, and we can as we are monitoring the temperature, we get to know whenever the temperature falls down or whenever the temperature rises too much, so we can turn on the water supply into the cooling jacket if the temperature increases, and this will prevent the enzymes of the microorganisms from being denatured. So let's write the answer. Monitor, it helps to monitor temperature to turn on water supply to cooling jacket if temperature increases to prevent enzymes being denatured. And with this, question number 6 ends. Moving to question number 7. Some mice have yellow hair and some mice have brown hair. The gene for hair in mice has 2 alleles. The dominant allele is H and the recessive allele is small h. A scientist crosses a yellow-haired male mouse with a yellow-haired female mouse. He predicts a 3:1 ratio of yellow to brown mice in the offspring. Give the genotype of the yellow parent and that would produce this 3:1 ratio. So I do not know how this will happen. So at first I will have to draw a Punnet's diagram. So I randomly took that both parents will be heterozygous. As I'm considering they both are heterozygous, so they will both have one capital H, one small h, one capital H, one small h. And that would create this, and this, and this. So this eventually means that this will be a yellow, this will be a yellow, this will be a yellow, but this small h will be a white, and this eventually indicates a ratio of 3:1. 3:1. So this means that both the parents were heterozygous. That is, they both have the genotype for them was capital A's and small a's. So it is H and H. The scientist then crosses 3 other pairs of males and female yellow mice. The table below shows these results. Calculate the ratio of the total number of yellow to brown mice for these crosses. So if you add 7 and 8 and 6 together, you get a value of 21. And if you add 3 with 4 and 3, you get value of 10. And the ratio would be yellow is to brown. And that is yellow was 21 and brown was 10, and you get a value of 2.1 is to 1. So let's write the ratio in here. 2.1 is to 1. And with this, part B ends. Moving to part C. Suggest reasons why the predicted ratio of 3:1 is not obtained. First of all, the sample size is small, like we used only 3. And fertilization is a random process, and mutation may have occurred during the fertilization, and we also got more number of brown haired mice than yellow haired mice, so the number is kind of equal, but the prediction is wrong. So let's write the answers. The sample size is small. Fertilization is a random process. Is just random. More brown hair mice than predicted. And mutations may have occurred. So mutations may have occurred. I hope 7C was clear, and with this, our entire paper 2 of January 2020 biology ends. I hope this entire video was easy for you all to understand. If you have any questions even after watching this video, you can comment the questions in the comment section, or you can email us at the Facebook page, that is Igcse, and the email address and the Facebook link will be all provided in the description box, and also the link for this paper will be given in the description box, and the mark scheme for the paper will be given in the description box, and the screenshots for the pages with answers of this video will be posted in our Facebook group or page, that is Igcse. And I hope it was clear enough for you all to understand. And if you like this video, please give it a thumbs up and subscribe to our channel. And thank you for watching.